Hello. In today's video, we will learn in detail about two important nucleic acids, namely DNA and RNA. By the end of this video, you will have a clear conceptual understanding of chemical components of nucleic acid, structure of DNA, semi-conservative replication of DNA, packaging of DNA, structure of RNA, types of RNA, and finally, differences between DNA and RNA. Nucleic acids are the building blocks of living organisms. They are the biopolymers of large biomolecules essential to all forms of life. In 1869, Friedrich Mescher first isolated a cellular substance from the nuclei of pus cells and called it nuclein. Since this material has acidic properties, it was renamed as nucleic acid. The functions of nucleic acid is storage and expression of genetic information. Nucleic acids are long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. These nucleotides store genetic information in biological systems. Deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, the principal genetic materials of living organisms, are chemically called nucleic acids. We shall start with the chemical components of nucleic acid. Nucleotides are the structural units of nucleic acids. Each nucleotide has three components: pentose sugar called deoxyribose or ribose sugar, phosphate group, and nitrogen base. Nitrogen bases are classified into purines and pyrimidines. Adenine and guanine are purines, while thymine, uracil, and cytosine are pyrimidines. Thymine is present in DNA and uracil is present in RNA. A combination of pentose sugar and a purine or pyrimidine base is termed as nucleoside. A combination of nucleoside and phosphate is called nucleotide. That is, pentose sugar, nitrogen base, and phosphate together form the nucleotide. In a nucleotide, nitrogen base is attached to first carbon of the sugar molecule by glycosidic bond. The phosphate group is joined with the fifth carbon of the sugar by sugar phosphate bond. The four bases, deoxyribonucleoside and deoxyribonucleotides of DNA, are as follows. The nitrogen bases are adenine, guanine, cytosine, and thymine. Deoxyribonucleosides are deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. Deoxyribonucleotides are deoxyadenosine monophosphate DAMP, deoxyguanosine monophosphate DGMP, deoxycytidine monophosphate DCMP, and deoxythymidine monophosphate DTMP. Let us study the structure of DNA. In 1953, James Watson and Francis Crick proposed DNA structure. DNA is a molecule composed of two chains that coil around each other to form a double helical structure. Each nucleotide contains deoxyribose sugar, nitrogen base, and phosphate group. The nitrogen base are of two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine and thymine. Purine base always pairs with pyrimidine base. Adenine pairs with thymine by 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. A DNA molecule is composed of 2 strands. Each strand is composed of nucleotides bonded together covalently between the phosphate group of one and the deoxyribose sugar of the next. The bases of one strand bind to the bases of the second strand with hydrogen bonds as shown in the diagram. The successive nucleotide of the same strand are linked by 3-5- phosphodiester linkage. As the base sequence present on one strand of DNA decodes the base sequence of the other strand, the strands are called complementary strands. The total number of purine bases is always equal to the total number of pyrimidine bases. This is called Chargaff's rule. By the way, if you are getting some value out of this lecture, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us recollect the important points learned so far regarding DNA. DNA is made up of 2 polynucleotide chains, pentose sugar, phosphate group, and nitrogen base. It is a double helical structure. The bases present on one strand pair with the nitrogen base of opposite strand. They are known as complementary strands. Adenine forms 2 hydrogen bonds with thymine from opposite strands and vice versa. Guanine is bonded with cytosine with 3 hydrogen bonds. Each strand from a DNA acts as a template for synthesis of new strand. Due to complementary base pairing, purine-pyrimidine ratio is always 1:1, which is known as Chargaff's rule. The 2 chains are coiled. The pitch of the helix is 3.4 nanometer and there are 10 base pairs in each turn. Each strand of DNA has a specific polarity. One end is 5' end and the other is 3' end. One of the strands run in 5' 3' direction while the other in 3' 5' direction. Now we will learn about the semi-conservative replication of DNA. The process in which a DNA molecule produces exact copy or replica of itself is known as replication of DNA. In eukaryotes, it takes place inside the nucleus. There is a definite region in E. coli DNA where the replication originates. Such regions are known as origin of replication. In eukaryotes, DNA replication occurs on multiple origins of replication along the DNA template strand. It takes place at S phase of the cell cycle. Initiator proteins initiate breaking of hydrogen bonds between the two strands. The DNA double helix is unwound by helicase enzyme. The DNA molecule appears as inverted Y-shaped structure called replication fork as shown in the diagram. Notice the leading strand and the lagging strand. Each separated strand acts as a template and synthesis of new complementary strand takes place with the help of RNA primer and enzyme RNA primase. It gets attached at the 3' end of the template strand. DNA polymerase builds a strand in the 5' 3' direction. On one strand, replication is continuous and is known as the leading strand, while on the other strand, it is discontinuous and is called the lagging strand. The discontinuously synthesized Okazaki fragments are later joined by DNA ligase enzyme. Enzyme RNase removes the RNA primer. Each copy contains one original strand and one newly synthesized strand. They undergo coiling and two identical daughter DNA are formed. As shown in the figure, each daughter DNA molecule has one original strand and one newly synthesized strand. Now we come to the packaging of DNA. In prokaryotes, the DNA in nucleoid is organized in large loops held by proteins. In eukaryotes, the organization is much more complex. Histone proteins, which are negatively charged, are rich in basic amino acid residues, lysines and arginines, which are required for DNA packaging. Histones are organized to form a unit of 8 molecules called histone octamer. There are 2 of each H2A, H2B, H3 and H4. The negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome. H1 histone, as shown in the figure, is present where the DNA starts wrapping the octamer and leaves it. The core DNA consists of 146 base pairs, and the linker DNA consists of 54 base pairs. Each nucleosome contains 200 base pairs of DNA helix. Having studied DNA in detail, we will now study about RNA. RNA is a nucleic acid found in the nucleus as well as in the cytoplasm. There are two main types of RNA. One is the genetic RNA, which acts as a genetic material in some plant and animal viruses, in bacteriophages, etc. Second is the non-genetic RNA required for protein synthesis. They are of three types: messenger RNA, ribosomal RNA, and transfer RNA. Let us study the structure of RNA. The RNA molecule is a single-stranded structure. Chemically, it is a polynucleotide molecule. Each ribonucleotide contains a pentose sugar, a molecule of phosphate group, and a nitrogen base. The nitrogen base of RNA are two purines, adenine and guanine, and two pyrimidines, cytosine and uracil. The four bases, ribonucleosides and ribonucleotides of RNA are as follows. The four nitrogen bases are adenine, guanine, cytosine, and uracil. The ribonucleosides are adenosine, guanosine, cytidine, and uridine. Ribonucleotides are adenosine monophosphate AMP, guanosine monophosphate GMP, cytidine monophosphate CMP, and uridine monophosphate UMP. Base pairing may occur between complementary bases as A pairing with U or U with A with two hydrogen bonds, and G pairing with C or C with G with three hydrogen bonds. Among the non-genetic RNA, let us learn about messenger RNA. The name messenger RNA has been proposed by Jacob and Monod. The messenger RNA is a single-stranded molecule that is complementary to one of the DNA strands of a gene. Messenger RNA is synthesized in the nucleus using the nucleotide sequence of DNA as a template with the help of enzyme RNA polymerase II. The process of making messenger RNA from DNA is called transcription. Messenger RNA carries message to ribosomes for protein synthesis from DNA. It accounts for just 5% of the total RNA in the cell. A sequence of 3 nucleotides on messenger RNA is called codon. Each codes for specific amino acid. The start codon or initiation codon is AUG. UAA, UAG or UGA are nonsense codon as they do not specify any amino acid. They are also called terminators as they indicate that ribosome should stop protein synthesis by translation. Translation is the process in which the sequence of codons on the messenger RNA strand is decoded. Accordingly, the amino acids join to form a polypeptide chain resulting into a protein formation. Next, we will learn about ribosomal RNA. Ribosomal RNA is a single-stranded structure folded upon itself. It is found in the ribosome and accounts for 80% of the total RNA present in the cell. It provides proper binding site for messenger RNA on ribosomes. rRNA combines with proteins and enzymes in the cytoplasm to form ribosomes, which act as the site of protein synthesis. Next type of non-genetic RNA is the transfer RNA. Transfer RNA is single-stranded and is also known as sRNA. S stands for soluble. It accounts for 10 to 20% of the total RNA in the cell. There are two models of transfer RNA. They are hairpin model proposed by Hogland and clover leaf model proposed by Hawley as shown in the figure. The anticodons present on transfer RNA are complementary to codons present on the messenger RNA. Like the hairpin transfer RNA, it has G nucleotide at 5' end and CCA nucleotide at 3' end. It carries specific type of amino acid at CCA end to the ribosome during protein synthesis. It arranges the amino acid in their proper sequence during protein synthesis. Having learnt about DNA and RNA in detail, let us now look into the differences between the two. DNA is a double-stranded molecule. RNA is a single-stranded molecule. DNA is mainly present in the nucleus, also seen in mitochondria and chloroplast. RNA is present in the nucleus and cytoplasm. DNA contains deoxyribose sugar. RNA contains ribose sugar. Bases present in DNA are adenine, guanine, cytosine, and thymine. Bases present in RNA are adenine, guanine, cytosine, and uracil. Adenine pairs with thymine and cytosine pairs with guanine in DNA. In RNA, adenine pairs with uracil and cytosine pairs with guanine. DNA obeys Chargaff's rule. RNA does not obey Chargaff's rule. DNA is self-replicating. RNA is synthesized from DNA. It cannot replicate itself. DNA transcribes genetic information to RNA. RNA translates the transcribed message for forming polypeptides. So, we have looked into DNA and RNA in detail. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video if you like and write down the questions. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.